just gonna get started now. How's everybody doing today? Good. You guys excited for science class this week? I think we're having a few more people join us. Yeah. Okay, awesome. Yeah. So for those of you guys who just joined us, we've just kind of started class. I'm just gonna run through a little bit about myself, how class works. If you have any questions, feel free to use the chat. I think my microphone's still working. My name is Viv. I'll be your instructor for these next 6 weeks. We're going to be doing, the class is kind of says in the title, but it's an intro to science. So we're going to be doing a little bit of intro bio, specifically like molecular bio and macro bio. So stuff like ecology and stuff like that. And then we're also going to be doing a bit of chemistry. So I've taught this course before. I've changed it a bit. I'm a biochemistry major in university, so I kind of changed my curriculum about that because I kind of want to get you guys excited about like things like PChem and OChem, which I know sound really difficult, especially since I don't know if you want to put in the chat, you guys are in middle school or not, but yeah, they sound really like hard classes, but when you get to university, you'll see they're not that bad. So I just kind of wanted to set you guys up for success for those courses. And of course, we're going to do like really general chemistry, but everything in science, as you guys may have heard, is like really interconnected. So I'm just going to introduce all those principles to you guys. And then finally, we're going to end it off with physics. So you guys are gonna be doing kinematics and E&M, but like the really, really basic stuff. So don't get worried or confused, especially if you don't like math or anything. I think actually math is quite fun once you get to physics. I think physics makes it a little more fun because you have to do more application stuff. So yeah, that's the plan. If you guys have any questions, again, you can email me, you can email our TA, Sherry. So Sherry, if you want to say hi and introduce a bit about yourself, can.
Hi, I'm a high school student in Washington State, and I'm the TA for this course. If you have any questions, you can message me in the chat either privately or to everybody, and then I'll be sending out like class material and homework after class each week.
So sounds good. So yeah, just to go over the general idea of what we have for the course for this summer session is that you guys come to lecture. I lecture for an hour and a half. It won't be me straight lecturing because I know that's really boring, especially when you get into college. It really sucks. So I will be asking you questions. I will be asking you questions like in between like slides to make sure you guys are still engaged. And then after every single session, I have a set of Kahoot questions, and those get asked the next week. So week 1 already has questions, but you guys aren't going to do it until the beginning of week 2. You guys get some sort of like refresher of the material. At the end of every session, I have application activities, what I like to call them. They're basically problem solving questions, and you guys can like throw up answers in the chat or unmute yourself. I'm totally fine with either. And we're just going to problem solve them together. And then I go over how I would have solved them. And these questions aren't just like you see at university, but I saw these questions when I was in middle school and when I was in high school. So it just kind of gets you like trying to get you prepped for like if you guys decide to take AP. I didn't do IB, but a little bit of IB as well. So again, just trying to get you guys like set up for success later on. There is an optional assignment that Sherry will send out to you guys after the course, like after the session is ended. And those are just like a mix of multiple choice, true and false questions, a bit of problem solving. If you guys choose to do them, I will send you guys the answer key or send Sherry the answer key and she can send it to you. We can also mark it for you and also just to give you feedback of like how we would have graded it, like if it was a high school type of question or like a college type of question, depending on what you guys are looking for. So without further ado, we'll get started. This week, we're going to be doing Intro to Basic Research Principles and Macromolecules. So basic research principles like scientific method, all that sort of stuff. And the reason I just bring it up is because like since middle school, high school, and college, we always cover it. And so it's always good just to have that general understanding because when you get into university, you use it a lot. Also, macromolecules, it's a really fundamental idea of how we can like connect things to chemistry from biology and so on and so forth. Also like to physics, like biophysics is also really important. So to get it started, what is this? Sorry, technical difficulties. I'm just going to ask you guys, what do you think science is and what do you think research is and how are they connected? You can put that in the chat or you can unmute yourself if you want to. If not, I can do my little spiel, but is there anyone who wants to say what they think it is? Okay, that's okay. So basically, also like if you have a question, feel free to speak up. Like I am totally okay with that. It's always good to get your question answered because I guarantee you there's going to be someone else in the like group that has the same question and everybody can learn from it. But literally science is kind of the pursuit of knowledge. Basically, if I boil it down to a really simple phrase, you're trying to figure out something that's happening in the natural and physical world. You don't really go something that's not within those barriers, like you have to focus in that specific area. So you're looking at behaviors, you're looking at structures. You can either experiment, you can do observation, and I will explain what the two differences are between that. And then like we use different types of reasoning, just to do a little bit of like a spiel on that. You can have inductive and deductive. Inductive reasoning is like you make a specific observation and then you apply it to a general population. So like if you had, for example, like a metal can, you say that can is metal, that's specific, and then you say, well, then all cans have to be metal. And then deductive is the opposite of that. So he says, well, I have all these metal cans, and that just means that this one single can has to be metal. It's like general specific. You can use both, either or, when you're making some sort of assumption or something when you're getting started in your research. So basically what reasoning allows you to do and what observation and what experimentation allows you to do is you have a methodology, which we're going to get to what type of methodology, to like figure out what type of science, what type of knowledge you're trying to find. So science is the pursuit of knowledge, research is how you get to that type of knowledge. And that pursuit isn't always linear, right? It's like a very creative process. I know some people will say, well, hard sciences are hard. And like, it's not like arts, where arts is like, you know, it's creative, you're doing stuff, you're making new things up. But science is a lot of that, especially like in chemistry and biochem, when you're trying to make something new and novel in terms of like pharmaceuticals and stuff like that. Like you need to have that sort of creativity, especially when you're trying to figure out new reactions and stuff like that. It's very important. And then again, going back to pursuit of science and stuff like that, you do need to have the ability to do predictions and explain. And so that leads us to the realm of like, what's the difference between a theory and a law? So a theory explains why something happens. It's been proved so often, not proved, we're gonna get to what that means, but it's been observed for so long, and that we can now understand a mechanism for why it occurs. So like the theory of general relativity, that's like a common theory, right? It explains why something happens. And then a law just like is different, because a law would explain, it wouldn't explain, it would just denote an observation and say, well, this would happen so many times. So how we figure out that would happen is we make a lot of observations, we do a lot of experiments, and then we say, well, this is common, so this becomes a law. So we have like Newton's third law, Newton's second law, all those like laws we're actually going to cover in kinematics. So it's just something like an observation, and you know it will happen. You don't explain why it happens, because that would be a theory. So they're two different things. So that's why I was just showing you guys with the images. So does anyone know what the first image represents, like in the top right? If you do, bonus points. I don't think we do bonus points, but you would get imaginary bonus points. You guys know what that is? It's okay if you don't. It's an X-ray diffraction of DNA. So back in the day when they were trying to figure out what the structure of DNA was, they had a bunch of different structures. So they were doing different types of reasoning, they were doing different types of experiments, and they were making structures. So they were thinking maybe it might be something called a triple helix. So it's a helix, but there's triple coils. But then they figured out it was a double helix, and they found it out with this method called X-ray diffraction. And so that's a picture taken by Rosalind Franklin. There's a whole sort of controversy thing about who figured it out, but we give credit to Rosalind Franklin, although Watson and Crick are kind of credited with the discovery of double helical DNA. In the bottom picture, that is an image of Newton's third law. So we're going to get to it at the end, but basically it's for every force there's an equal and opposite reaction. So as an example, you slam a wall with your hand kind of hurts, right? Because the wall is doing an equal and opposite force on your hand, but your hand is smaller than the wall. So yeah, it hurts. Not that you should try doing that, but that's what happens. So then research. So we covered what science was, right? It was the pursuit of like learning stuff, of studying behaviors, of studying structures. Research is how you do it. So you have to do research to answer your questions. It like along the way, you can learn how to do different things. You can like make discoveries in technology, medicine, even other types of research. Sometimes people like will try to do something, one field of research, and then realize they made a methodology, they made a technology that's actually more applicable for a different type of research. And so it's kind of beneficial in that aspect. You can do it, like I kind of boiled this down, but it's achieved through observations and experiments. And then I'm going to explain what different types of observations and experiments you can do. And the most basic methodology of getting through a type of research is scientific method. So I'm pretty sure most of you guys have heard what the scientific method is or kind of know something about it, but we're still going to go over and review it just so that everybody's on the same page. And then finally, research is like not a linear process. I think the most important takeaway from research and science is that nothing's linear. So nothing's going to have a start and a finish, like in a timeline. It's not going to be that way. It's always going to be very cyclical. So if something goes wrong, you always have to go back to the beginning. Or if you get success at the end, you still have to go back to the beginning because you can always learn more and you can always do more experiments. And the reason why stuff can be cyclical is that you have to have peer review. So what peer review means is that, let's say you make a discovery about something or you do some sort of reaction and it was just you yourself doing it. But who knows if it was actually true, right? You need someone to verify that it's true. So we have peer review where people will either go over your research paper that you produce or actually or replicate everything that you've done. And so when they get similar results or a similar observation, then they will say, well, yes, this is indeed like suggesting that your work was actually correct, or supported whatever you were trying to like observe in the beginning. So there's a bunch of words that we have to be careful when we're using with research. So it's just kind of hard to like get the vocab right. But yeah, peer review is very important. So the scientific method is like a set of principles that we use a lot. We always say linear, like when we're first teaching you guys in middle school, it's always like it's a linear process. You have a hypothesis, well, you have a problem, then you have a hypothesis, then you have some sort of procedure, materials list, then you do the whole experiment, and then voila, you have your conclusion, or however you want to say the last part. But you get that to the last part, and you conclude whether or not it was supporting whatever your hypothesis had or rejected your hypothesis. But a lot of times when you progress later on through education, you're going to see that it's not like that. You have a lot of like speed bumps and sometimes you just have to restart, and it's a whole lot of fun. But basically it's a lot of repetition. You just got to keep doing it until you get it right. Sometimes you don't know what right is and you just have to do it and you'll just get a bunch of different results. And that's important in science because then you have a lot of different like not answers, but different options that you can use for like further research branch points. So your general steps, really basic here. You have a problem, you can observe, and then you observe and you describe it. Then you make a question. In science, we call that a hypothesis. So it's typically an if-then statement. When you read research papers, a lot of the times they don't use if-then statements. If-then is like a really basic way for people, like students, to understand this is what a hypothesis means, but later on you'll see it's more like an actual question. So they'll be asking, how can this like happen? Why does this happen? And that'll be more of their hypothesis. And then you come up with a whole methodology to test your hypothesis. And that depends a whole lot on what you're trying to look at. So we'll look at observations and experiments. Also, then that brings up the question of what statistical tests you want to use. And we'll explain why statistical analysis is really important in terms of research. And then you draw your conclusion based off of your data, based off your statistical analysis, and then you also send that off to peer review. And so that's why I say it doesn't have to be a last step, because you could show that your hypothesis is not supported, and then you would have to form a new one and then go through the entire experimental procedure again, like a different one, and then see if that supported the hypothesis. And then you conclude and then peer review and all that good stuff. Or you could make sure, or you get a result that said that your hypothesis is supported, and that's good. So then you can move on to more research to back that up, or you could start a whole different procedure methodology. It's up to you, but that's kind of like the gist of the scientific method. So I keep saying it's not a linear process, and it is very much a cycle. And yeah, peer review is always important. And then the most important part besides peer review is that you always have the ability to refine and change your hypothesis. In doing so, you can actually get a little more specific research question, and then it can allow you to figure out a more specific procedure. And so in research, we like to do something called law of parsimony. Basically, that's just trying to figure out what's the most basic procedure to use, and that gets you the result, not the easiest, but the most simplest design. So that way you're not having a bunch of different variables that are kind of like hurting your result process. And so I'm going to explain what that means later on. But basically, the gist is you want to be able to have something that has a refinable hypothesis, something that has a very simple design when you're making your procedure, when you're testing it, something that allows for you to do easy statistical analysis for the most part. Sometimes you can't really do an easy statistical analysis. I don't know if you guys have done any sort of, it's called inferential statistics. If you haven't, I'm going to describe a little bit about it. But you kind of want like the simplest method of doing that, because sometimes it can just get so convoluted and so difficult. And then finally, you want something that's easily peer reviewable, so that when you're explaining to people what you've done after you've done all that research, they understand what you're talking about. Because you could have done all this research for years and put out something on like one sheet of paper, but it's not really peer reviewable because no one knows what you're talking about, and then they'll dismiss it as, oh, you're crazy because you did all this work and now it's on one page and it doesn't make any sense. So you want to make sure that it's in a format that people understand and can also replicate. And that's the other important thing about science is we do a lot of things with replication because we need to be able to show that this was an actual observable effect. So experiments versus observations, and then I'm going to like explain which one is better. It depends on what you're using it for, but sometimes there's one option that's better than the other. So experiments. I'm just doing really basic stuff here, but in general, they're used for cause and effect relationships. And you're basically manipulating a variable. We call that the independent variable. You pick something and then you can either have it on or off or more or less, something like that. And then you're measuring the output from that change, and we call that the dependent variable. And we use control groups to show that if something was kept the same in its natural environment, what would happen if we change something. So you have something at least like as a basis to compare it to. So this is where replication comes in as like an important factor. Also sample size. So replication allows you to make sure that if you observe something once and we thought, oh, that's weird, if you have it done multiple times and you see that same weird effect, then you know that, okay, we have something here. Otherwise, you have this thing called an outlier, and then that's not really good for research because it was just a one-time thing when everything else was different. So then that means your conclusion, if you made that one conclusion off the outlier, would be like incorrect, and you're not quite sure if you would have to redo your entire procedure again. And then that leads to a whole different mess when you start talking about statistics, because it just kind of ruins it. And then the other thing besides replication is sample size. So you want to have a lot of samples when you're doing your experiment, because then you have some form of replication. Basically, it's not exactly replication, but you just have more instances of stuff being changed. And that also helps for when you get to the statistical analysis part of things. And then in experiments, we always try to pick things that are not going to confound or mess up the rest of the design. So we call those variables confounding variables because they confuse and mess up the design. So you don't want something that has like an ambiguous effect. So the same, like a really weird, confusing effect that might hurt what you get from the dependent variable or might change the independent variable in some way. So it wouldn't be working to its full capacity. And then this is where we get to the point of the parsimonious design. it's just saying it's a simple design. So we want the most simple sort of design for the experiment so that we don't get confounding variables and that we have enough replication and enough sample size to do really good statistical analysis so that we can make a good conclusion. Does that make sense so far? Yes? Also, if I'm going too fast, just say so. Does that make sense? Anyone? Okay, because I'll take that as it's okay. So then for, I was going to ask what the independent variable was for this diagram, but that's okay. It already tells you. So yeah, for this diagram, I just wanted to show you that you do have an independent variable and you do have a dependent variable. So in this case, the independent variable, the one that's being changed, is the amount of water. So the control group in this case is no water is being added, and your experimental group, it is being added. So then you measure how many seeds sprout afterwards, after the seeds have been planted inside those pots. So that's dependent. And then like in the typical experiment, you would have a lot of pots, and then you could just measure and making a conclusion after looking at every single control pot, after every single experimental pot, and then you would have enough data to do more statistical analysis. So this is what I mean about wording for experiments. If you get a conclusion that seems correct, it's not correct. Because it can't prove anything. You just have something that agrees with a general trend or observation that you made initially, and it supports what you thought of initially. So that's why we say it supports. So if we got a conclusion that looked good, we don't say it proves, we don't say it's correct. We just say it supports our hypothesis, and then we move on with our future research. If it doesn't support our hypothesis, if the data we get back is like really off the charts and it just doesn't make sense to what we wanted, like we thought about initially for our hypothesis, we say it rejects it. So yeah, don't say prove, don't say disprove, don't say correct or incorrect. It's always support or reject. If that's the best way to think about it, because when you start getting into the realm of like saying it proves this, that's kind of like a law theory sort of situation, and it's not really feasible when you're just doing one experiment with like one go of an experiment. Like you would have to do so many experiments to show that it would prove, and it doesn't really, like it just gets confusing after a while. So then experiments are like different from observations. Observations, you don't change anything at all. So you don't have any independent variables. You do have a dependent variable because you're trying to measure stuff, watch behaviors and all of that stuff. And there is a good point and a bad point to using observations. So observations are good when you're trying to watch like something that's happening in a natural habitat, like you're trying to count the number of wildebeest or something in the Serengeti or something like that, where you're not having to change any sort of variable because there might be ethical reasons of why you would like not do it. So there's a lot of research like that on humans that's like observation. You don't change anything because if you did, that might land you into some legal trouble. On the other hand, experiments are good when you need to know if there's a change in something that would get a better result or get a different result from what is naturally observed. So we see a lot of that when we're trying to experiment and figure out what reaction is good for like a pharmaceutical drug and stuff like that, or what happens when we have this bacteria that has this sort of, we call it a plasmid. Basically, it's a DNA package that gets inserted into the bacteria, and sometimes it can confer or give an advantage to the bacteria to make it live longer or survive some sort of antibiotic resistance sort of thing. Like if there's something that might kill it in its habitat, like if it's in a petri dish and there's like some sort of antibiotic in it, it can survive that. So that's why it has antibiotic resistance. So like there's different experiments to see what's beneficial, what's not beneficial, things like that, but it wouldn't affect like a human or an animal or raise legal questions or something like that. So that's when it's good to use an experiment versus an observation. Does that make sense? Chat. Okay. Sorry, I'm just like reading. My computer's like really bad. I think, Sherry, we're providing a video recording, right? I think. Not sure.
Yeah, that's correct.
Yeah, okay, so yes, I didn't mention that at the beginning because I forgot. Sorry, guys. But yes, if you have a class at some point between now and like the end of class, yes, we will be providing a video recording. So yeah, sorry about that. So we're just like going on about how experiments work. So experiments, like they're helpful in certain cases, right? Especially like when we had back in the day, when we had like big debates on about like what was the method of generation. Like in this case, it was spontaneous generation. So a little bit of background history. Like spontaneous generation was this like idea, phenomena thing that happened in 350 BC. It was supported by a lot of philosophers because like how could you explain why, for example, if you had a piece of meat or something that just stayed out wherever, like the next day there'd be like maggots on it, like baby fly, like the larvae, you guys know what that is. So like they were wondering, how does that happen? Do these things just come from air? Where do they come from? So at the time they thought they just kind of like poofed into existence, but now we know that's not really the case, right? It'd be kind of cool if that happened, but not so cool if it was like baby flies, right? But yeah, anyway, this phenomena thing got rejected in the 19th century by this guy named Louis Pasteur. Louis Pasteur actually did a lot of cool stuff. He didn't just do this one experiment. He has a bunch of different things like pasteurization. You guys heard about that if you guys drink milk and stuff like that. He did a bunch of cool mechanisms to like in different fields, like a lot of things in chemistry, a lot of things in biology, and it's just pretty cool to read about him. Anyways, so he wanted to like reject, not just prove, he wanted to reject spontaneous generation. And what he did was use these things in this picture called swan neck bottles. And basically what they are is they're just like little kind of like bulb bottles, and then they have this really thin neck that comes out into like a little almost L shape. And so you can allow air to come in, but whatever is in the air, like spores and stuff, gets trapped at the bottom of the like the neck bit. So air can come in, but like bacteria and stuff that's floating in the air can't come into the broth. And so what he did was he sterilized broth, so like beef broth, soup from meat and stuff, and he sterilized it by heating it to a boiling point, and then he put it into the swan neck flask, and then he used an open neck flask, which is it's supposed to be this one. This wasn't a good picture, but open neck just means it didn't have like the little swan neck at the top. And so he left it for a period of days, and then he noticed that in the open neck flask he had bacterial growth, and then in the swan neck he didn't. But when he did like swish the neck around to allow the bacteria that were caught within like the little L trap, a bit of the glass to come into the broth, it actually did grow bacterial growth inside the broth. So then in that case, he showed that in that case for his hypothesis that it did support that the air does carry spores and bacteria that would lead to growth, like it wouldn't allow necessary for spontaneous generation to occur. So in this case, his control was the open neck flask because that's what it was. Like that's what they had lying around. Like a swan neck flask is kind of weird, and like no one really has that lying around. So that was the independent variable. They were changing the shape of the mouth of the glass. And the dependent variable was the amount of bacterial organism growth shown in the flask. So that's how they kind of rejected. Well, that's how they rejected spontaneous generation. They didn't kind of reject it. And there were a bunch of other experiments that did the same sort of thing. I think like Francisco Redi did something like this, but with like meat and cheesecloth. So, but the principle is the same. Like they all had a control, they all had a dependent variable, and they all had an independent variable. And then they just did it a bunch of times, and they just showed, well, this isn't the case. So the experiment is the fun part. You get to do all this cool stuff, use all these different techniques, get to make all these cool observations. If you're doing reaction stuff, you get to see all these different color changes and all these different crystals. It's really fun, it's pretty. But then you get to statistics, and then that kind of isn't, depending on the person, it is the fun part or it isn't the fun part, because there's like a bunch of like different things you have to do to make sure that you get it right the first time. So statistics allow you to make predictions based off the data. And this is the main part that allows you to say whether or not your hypothesis was supported or rejected. So you can't necessarily say that your hypothesis was rejected or supported when you first did the experiment. You have to go through all the different types of replications that you did in the experiment, and all the different like you want to make sure that your sample sizes are the same when you do the experiment to have some sort of consistency, because again, we want the most simplest design, right? So when you get through all of that data and you analyze it, you want something that can allow for easy interpretation of your data. And statistics is important because we use that to explain a lot of data to a big audience, right? Because some people don't really like learn statistics in school, or that's not their focus, right? So you want to make sure you have something that's easily interpretable for people who haven't really studied it, or like for everybody basically. So you have two different types. You have differential and you have descriptive and inferential. I'm thinking calculus. You have descriptive and you have inferential. So basically, I'm going to explain what it is like in the next few slides, but descriptive is kind of like what you see at this bottom part of your screen. You have something that's describing what's showing like inside your data, what is your median, what's like the central tendency, what is your spread of your data, sort of thing. Inferential is when you're taking like a sample data and then you're inferring what the rest of the population that it came from looks like. So we use something when you get into like university, I think they might even do this in high school at this point, but they use this thing called R. So it's a type of coding language, and it allows you to make it allows you to solve for different statistical tests. It's pretty handy. And then this picture down here is something called a chi-square test. It's something that we use commonly in inferential statistics. Basically, what it allows you to do is it allows you to do a comparison between your observed data and your experimental data. And so from there, you get this thing called a chi-square value, and then you can use that value to say whether or not compared to, it's a whole step, but the main point of the whole process is once you get that chi-square value, you compare it to a different chi-square value that's known, and then you can make the assumption or make the observation that either your hypothesis was rejected or it was supported. There's a whole step. I kind of just like boiled that down to like 2 things, but it's a whole step. You guys can look it up if you guys are curious. I think I put a link in the slides afterwards, but does that make sense to everybody? Sort of? Kind of giving like the general spiel on it, but if you guys have any questions, please let me know. Anyway, so for descriptive statistics, like I said, it's really basic. It's just working with what you have. You are not inferring anything, like you can't infer anything from it. You're just showing, well, this is what I have. This is the mean, this is the median, mode, my different quartiles. We use something called a box and whisker plot. You guys probably have seen that. And that just shows the distribution of data. So basically, descriptive statistics can tell you only things about the tendency and about the spread. So where things are located. So like the median is always going to be the middlemost data point. The mean is going to be the average, and mode is the most frequent number or data that shows up. And then range is from your min to your max. Quartiles are little percentile bits that are within those different ranges. And then standard deviation is kind of like a spread of where things are compared to the other, and variance is standard deviation squared. I think it's been a while since I've looked at the formula sheet, but it's another description of spread. And then we have different descriptions of what skewed is. Like we always say there's, it's positively skewed or negatively skewed. Basically, skewed just means how far data is pulled up or down based off like what's the highest or lowest number. So this is negatively skewed because you still have negative numbers pulling it back, and the positively skewed is like you have really high numbers that are pulling the curve forward. And then you have something called a normal distribution, which I think you guys may have heard of. It just looks like a happy upside down U. It's like very symmetrical. So in a normal distribution, in a normal distribution, everything's lined up, positive skewed, some stuff's dragged forward, so your mean is going to be higher, and then negative is going to be lower for your mean. But yeah, I think they ask questions like that on the SAT, probably, just for you guys' future reference. But yeah, for inferential statistics, like I said, you're taking data from a like one single sample or multiple samples, and then you're generalizing that behavior to the whole population that it came from. So that's what this diagram is showing. It's showing that we have this big population, and that we have these individuals that get sampled, and there's like so many techniques that you can use to get those individuals. I didn't list them because it's a lot and it's kind of difficult to explain some of them sometimes, especially when I don't have like a good diagram, but basically the point is you get these different individuals and then you do whatever experiment, observational study, survey, and then you make those like not assumptions, but generalizations to the main population. And these don't have to be people, by the way. This was just a good diagram that I found that kind of helped explain it. So the reason we have inferential statistics is because when you're doing an experiment, you always have error. So inferential statistics allows for like a little bit of leeway and a little bit of like, not help, but it just helps your like statistical analysis to cover for that error. It won't cover for all your error, but it's just there to say, well, we do have error, but this is what else we have from this experiment. So in inferential statistics, it's not like descriptive where we only had central tendency, central tendency and central spread. We do something called hypothesis testing, and there are so many different types of hypothesis testing depending on what your experimental design was. For example, if you had 2 samples or something, or you did some sort of weird group test thing, it depends. Like if you Google it, you can see all these different tests for different types of experimental methods. And if you're doing research, you do have to know or have a gist of what type of testing goes with your methodology, because different tests will yield different results, and you want to make sure that your tests line up with what you had in the beginning, especially because it has to do with a lot of your replication and a lot of your sample size. So that's why we say sample size has to be always kind of big when we do inferential statistics, because if you only have 1 or 2 samples, that doesn't necessarily help your case, because it's so few compared to like if you had a sample of 30 or 300, because behaviors in individuals are always going to be different. And so the difference between 1 and 2 individuals is going to be really drastic compared to if you had 30 or 300, because then there might not be a difference if you had a really big sample size. So for hypothesis testing in general, though, you do start off with a null hypothesis and alternate hypothesis. So the difference between those two, it's kind of like a true and false sort of thing. So a null hypothesis says something is going to happen, and an alternate hypothesis says something is not going to happen, and then it's up to you to support or reject your null hypothesis. And if you reject your null hypothesis, that means you support your alternate hypothesis. So it gets kind of confusing in the wording, but that's kind of like the gist behind the different hypotheses. It's basically a standard for what you can work off of. And then you use something called, like you can use 1-2 tail predictions. You can do different statistical tests. So like I showed you the chi-square test. That's if you wanted to compare between observed and expected. And then you use something called a significance level. And basically what that is a standard that's accepted by all scientists. It depends on which type of significance level you want to use, but it's accepted by most of them to have like a standard to say, well, this is where we draw a line of saying this is rejection and this is where we support the null hypothesis. So yeah, and then I just say that for the null hypothesis, not to confuse you guys with how you would word it for the alternate hypothesis, but just know that if you reject one, like you can't reject both. You need to reject one so that you can support the other, and you need to support one to reject the other. That's just how it works. It's never both. So that's the gist. You need null and alternate hypothesis, the hypothesis test, then you need to look at a significance level. In some cases you get a p-value, but you use that to compare with significance level, and then you can make the decision to reject or reject or support your null hypothesis, and thereby you decide what to do with your alternate hypothesis. And normally it's really clear cut because inferential statistics uses tables to tell you what value works for your rejection or supporting of the hypothesis. What I mean by table is like you have these preset values that have been calculated by statisticians, and then you don't have to do these, you have to do calculations, but you just need to make sure that your value is above or below these values, and then that's how you do it. Does that sort of make sense? You guys are really quiet.
Yes.
Yes, yay. Okay, awesome. Someone. Cool. So that was all of the statistics questions. So I'm just going to ask you statistic topic. I'm going to ask you a couple questions. You guys can answer it if you want to, or I can just kind of explain really quick. So does someone tell me what's the difference between descriptive and inferential statistics? Wait, Oh, what grade is this? Oh, yeah, actually, I did statistics in middle school and high school. So I did like 8th grade, and I think I did this 10th grade, and then they make you do it in university. So yeah, a lot of fun. Yeah. But I think every school is different. My science teachers made me do this because they wanted us to understand why, like if we were looking at something, some sort of research paper or some sort of problem, that we'd understand what the question was asking. But yeah, no, descriptive statistics, I know it's like a lot of information, but descriptive statistics is like you get data from a sample and you don't infer anything off of it. You just look at what's my most central values, what are my spread values, and that's all you get. Inferential statistics we use to make generalizations about a population. And then we do all sorts of crazy hypothesis testing. That's the only difference. I think inferential statistics is just a bit harder in that aspect. But does that make sense to everybody? Yeah. Okay. And then true or false, is scientific method linear or not? Well, I guess yeah, is linear. That was a really bad true and false question because I just made it up. But yeah, true or false, scientific method is linear. And I have to like hear myself again. I said scientific method is linear. It's not linear. It's actually, yeah, it's false. Yeah. So just to recap on that bit, yeah, the scientific method, you can have a lot of points in the method that where you're gonna need to like restart or change something. So it's not linear in the aspect that you go from hypothesis all the way to conclusion, because something might go wrong in your experiment or something might change in your statistical analysis where you have to go back and refine your hypothesis. And then also when you get down to your conclusion, you also need peer reviewing. And peer reviewing might actually send you back to your hypothesis because what you may have done is not replicable for other people. And so your data kind of is just weird and it doesn't make sense. Like it just kind of poofed out of nowhere. And that's not good in the scientific world, because if we can't replicate it, then it's kind of like, it's not, you can't support it. You can't support the existence of that data. So it's just a question of where did you get that data? It doesn't make sense. So that's why it's not linear. Does that make sense? Yeah. Okay.
Yes.
So that, okay, awesome. So that's the gist about scientific method and statistics. If you guys have any questions, please let me know. I know it's a little overwhelming, especially since I kind of like barreled through it. But if you're confused, again, please let me know or let Sherry know. So now we get to do macromolecules, which are really fun. Like I like them a lot. They're pretty cool. So I know this is the biology aspect of things, but it kind of dives into the realm of biochem. But don't worry, you guys can say you did a little bit of biochem today. It's not that bad, I promise. So common elements that we talk about in biological systems. I just want to double check. You guys have talked about elements, right? Yes, thumbs up. Yes, no, chat. I think I'm just gonna assume you guys have. I'm going off like old notes that I had, but basically, we have a bunch of common elements that we see in biological systems. Those are carbon, hydrogen, oxygen, nitrogen, and phosphorus. Yeah, in school. It depends on, I guess, where you've gone to school. Sometimes I have students who've seen it and sometimes they haven't seen it. But elements, they're just substances that we have in the world around us. And we've basically condensed them to something called a periodic table. And those elements that are on the periodic table are pure elements, and sometimes they're like building blocks. You can take them together and you can make molecules, which are combinations of those elements, and the subbits of elements are called atoms. So a type of element has atoms, and so atoms are in everything, right? Like you and I are made of atoms. We're made of molecules. Atoms make up molecules. So that's kind of where I've been going with that. Does that make sense? Yes. Okay. I will try to, if you guys have questions, please speak up, because I'm just kind of like, I think they're okay with this. But yeah, you have carbon, hydrogen, oxygen, nitrogen, phosphorus. You guys probably know what H2O is. That's a molecule. That's water, right? So it's 2 hydrogens. Yeah, water. Good job. Yeah, it's 2 hydrogens and 1 oxygen. So when we get to chemistry, I'll explain why we say H2O, and it's actually kind of cool. But these different elements, like I said, have different things called atoms. So atoms make up, well, elements are like the big name, and then underneath it we have atoms, and atoms are little bits of elements, a pure element, and those can bond. So they share electrons with, share or steal electrons from other atoms, and those are called bonds. And when they do that, let's say H2O with oxygen is sharing electrons with the hydrogens. So that makes water. So that makes a different combination. And so we call that a molecule. So when we have these elements making these combinations in a bunch of different ways, like water is a molecule, but I'll show you when we get to sugars, lipids, and all that good stuff like fats. Those make something called macromolecules, and macro means big. So these are big molecules, and we, like you and I, we're all made of macromolecules. Everything we eat are made of macromolecules. Some of the stuff we wear is made of macromolecules. So macromolecules are really important because if we understand the structure, we can understand like what type of drugs we need, like if we're sick or something, what might kill bacteria if we have like a bacterial infection or something like that. It's like all very, it all ties in because all these different macromolecules all interact with each other. So it's important to understand their structure. So yeah, these, just to clarify, these little atoms, these are called atoms. This is a structure you guys, I think, learned in middle school. It's called a Bohr model. If you haven't seen it, don't worry about it. It's okay. A Bohr model isn't actually a true representation of what an atom looks like. I'll explain what it means in like the chemistry unit. But the Bohr model just shows you that you guys know the different components of an atom, right? Or I can do it really quick right now too. But yeah, an atom, like we'll focus on this carbon bit here. Actually, I think I have a better diagram on the next page. Oh, I do. Yes. So just to review for you guys, or if you've never seen this, don't worry, I'll explain it. An atom has 3 bits, like major bits. It has other bits, but we only focus on the 3 bits. An atom has something called protons, so it's these positively charged units, and then it has neutrons, which are neutral. So it's kind of easy to understand, right? So positive, proton, neutron, neutral. And then we have electrons. Electrons, I don't really know a good way of making it sound negative, but they are negatively charged. And so electrons are what we talk about mostly in chemistry and in, oh, I covered that definition, but in chemistry and biology when we're talking about electronegativity. So that's what we're going to talk about. Basically, electrons allow for bond formation. So it allows the ability of one atom to connect with another atom. And it can either do that by completely stealing an electron and then makes it an excess of negativity, right? Because if we said that electrons are negative, if an atom that was neutral, it had no charge, right? Because the positive charges here will balance out the negative. That this is a neutral atom. By the way, if it's like 6, I think it's 7. Oh yeah, 7 is nitrogen. If there's 7 positrons, if there's 7 protons, then there has to be 7 electrons for a neutral atom. But if there's 1 extra electron, that means it's negatively charged because you have an extra negative, right? Because that's like a -1, right? Because now you don't have 6 minus 6, you have 6 minus 7. Does that make sense? So that's negative. But in other bonds, you have equal sharing. So that's what this diagram is showing. So I'm just going to explain what these all mean, and I will definitely explain this diagram more if you guys have never seen this before. So electronegativity is the ability of an atom to take electrons for itself, like it wants it. So I can't remember what the most electronegative atom is. It's no, fluorine. Sorry, the most electronegative atom is fluorine. So they want electrons really badly because if you look at a periodic table, I don't know if you guys again have covered this, but you have something called noble gases, and noble gases have something called an octet. That means they have a full deck of electrons that makes them really, really happy. And fluorine is close to it, but it's missing one, and it really, really, really wants that one. It wants to join the noble gas club because it's the cool club and they have everything, but fluorine is missing one thing. That's the best way to think about it. It just wants it. So that's why it's the most electronegative. It just wants that one electron. Then you have electrons that want to give up their electron because they also want to be in the really cool electron having club, but they have one extra and they don't want that one extra. It's too much baggage for them. That's what electronegativity is. So electronegativity means they want to attract electrons, and then there's something called electropositivity, which describes the other part. But in most cases, we just talk about electronegativity. And that's really important for understanding reactions, by the way. If you can understand the ability for an atom to want an electron, you basically understood like the first step of organic, like OChem. Like that's the gist. But so then we have these different types of bonds. So we have covalent bonds and we have ionic bonds, dipole-dipole, and hydrogen bonds. I'll explain what that all means. So covalent bonds is sharing of electrons. So if you look in the diagram at the bottom, that's the first and second diagram picture of the thing. So when they're equally shared, we call that nonpolar covalent, because the two atoms, when they're forming that bond, they're pulling on each other's electrons, but they're pulling with the same force. So it's like having two people in a room of like equal force pulling a rope between them. They're not really going anywhere. Like no one's getting more rope than the other, right? That kind of makes sense, right? But polar covalent bond is, let's say we have a person who's stronger than the other person, like way stronger, and so they get more rope than the other person. So when they have an excess of rope, they get more rope, right? So like in this case, it's a polar covalent bond, they get more negative charge because they can get more of the electrons towards them. So that's why they drew it in like an electron cloud, which is also good to think of because electrons aren't like on a bead on a string, they're like in the cloud. So if you get more of the electron cloud towards you, you have more of a negative charge. Does that make sense? Yes? Good. I hope that makes sense. If it doesn't, please like say something. And then when it gets to the point that it's beyond polar covalent, when that person who's pulling the rope gets all the rope, that's an ionic bond. So when you have someone who doesn't have the rope and someone with all the rope, that's ionic because you have like a real, like they actually have the rope now, they own it. So the negatively charged, that's what it is here. The negatively charged atom here has the electron, it has the rope. The positively charged atom here, it doesn't have a rope, it's missing a rope, it wants it back. So what happens is this negatively charged atom, geez, I'm forgetting words. Negatively charged atom sees this guy and it forms a bond because it's stable that way. Because if you just have a bunch of plus things floating around, it's not stable. If you have a bunch of negative things, it's definitely not stable, right? Because I think a really common idea that we talk about is negatives attract positives, right? So that's what the idea is getting to. So that's an ionic bond. Then we have dipole, and hydrogen bonds. There's the same thing. Hydrogen bonds are just a subtype. So this is a hydrogen bond. What it is this sign here is called a delta, and delta negative, it means partially negative, and this is partially positive. So it means that this has a lot of electron density and this doesn't have a lot of electron density. So it's not quite ionic, but it won't get to be ionic, but it's just showing that this is way more negative than this is. So what happens is this gets really positive, and this is a hydrogen bond. This gets really positive. So then it sees the oxygen on this water molecule sees this guy, and it makes this really weak bond. It's not like a handshake. It's just like it sees it and it's trying to like touch it, but can't really touch it, but still makes an interaction. That's the gist of it. And then a dipole is similar to it, but it's more general. So yeah, that's the gist of it. If these were two different molecules, it would be a dipole. The only reason I bring that up is because when we talk about different structures, sometimes I'll say dipole, and I just don't want you guys to get confused, or hydrogen bonds. So this again is delta negative, delta positive. Delta negative means excess of electron cloud towards the oxygen. It's still sharing the electron cloud with the hydrogen, but most of it's on the oxygen. And this is like for here, they show it in a charge diagram. So this is a hydrochloric acid. So you have a hydrogen and a chloride. The chloride's more negative, and I'll explain how you can figure out what's negative and what's positive in the chemistry unit. So this just means that more charge density is towards the chlorine than it is the hydrogen. And then this is how we tell if something's ionic. So if there were 17 protons inside the nucleus of a chlorine atom, and there's 17 electrons on the outside of the nucleus, then we say there's a 0 overall charge, right? Because we said protons are positive, electrons are negative. So 17 minus 17 is 0. But if we had 18 electrons, that'd be 17 protons minus 18 electrons, and that's a -1 overall charge. So that's the difference between a partial charge versus a full-on negative charge. A full charge gets an extra electron or loses a whole electron, but a partial charge is just one's being shared and one atom is hogging it. One atom is taking it more for itself. Like it doesn't really want to share fairly. It's like, fine, I will share with you, but I don't want to share fairly. That's all it is. So we have 4 different classes of macromolecules. We have carbohydrates, lipids, nucleic acids, and proteins. You guys will, oops, I did not mean to do that. You guys will cover this, like, pretty sure, like middle school, definitely high school. And if you guys choose to do science in university, you do it a lot. But yeah, I'm gonna briefly explain what each of them are, just so you guys get a little bit of an introduction. So this guy is a carb, and I'll tell you how you can figure out which one's which. But this is a carb nucleic acid because this is DNA, right? This is a protein, and this is a fat. This is, yeah, that's a fat. Looking at it. So carbohydrates, they're sugars. You eat a lot of them. They're in like paper, plants. You use it for storage, like glycogen. You use that for yourself. Glucose, lactose. If you guys drink milk, you take in lactose. Sucrose is table sugar. You eat that probably if you like drink tea and stuff like that, or bake a lot. The common formula for it is, this is supposed to be a C sub X. So carbohydrate is named because there's a carbon and there's a water molecule. And the X just means a number. So C6H12O6, that's glucose. So, or a form of glucose. And the 6 is just multiplied to everything. So 6 here, 6 times 2 is 12, 6 times O is 6. Well, 6 times 1 is 6, not 6 times O. That'd be weird. But yeah, carbon and water. We have building blocks. Everything in biology has building blocks. So carbohydrates, building blocks is something called a monosaccharide. And that's just a single sugar. And the cool thing about carbohydrates is that they come out linear like this. This is called linear. But they can actually form rings like this in aqueous structure. So like you yourself, you're made like of water, right? So mostly water. So that's what happens to your sugar. Your sugar is not like a linear form. It's like actually aqueous. But yeah, so then you have something called a glycosidic bond that's used to connect your sugar between each other. So this is called a glycosidic bond. And then make something called a polysaccharide. So it means many saccharides. Does someone have a question? I thought I heard. No? Okay. But yeah, carbohydrates are typically used for energy and structure. So like I said, glycogen is a storage molecule that you have in your body as like a form of like storage. So then when you need sugar, your body breaks down glycogen, releases it to free form glucose. So then you can use that. If you drink milk, it's lactose, something like that. Structure, plants have something called cellulose. I think you guys have heard of that. We can't digest it, but like we use it as a fiber source. Cool thing about carbohydrates is that they can be modified. So like you have some, and I'll get to this in chemistry, but there's something called functional groups. Actually, I can describe it now. Functional groups are like these things here that's been circled. They're entities on the molecule and they give them really cool properties or different properties, something like that. So this is a functional group. This OH is a functional group. So this is called an aldehyde. This is called an alcohol. So if you're wondering, like when people say ethanol and stuff like that, ethanol has an OH and that's why we know it's an alcohol. And carbohydrates can also be modified with something called peptides, which are parts of proteins. And that's actually really cool because we see stuff like that in bacterial cell walls. And that's how we can use different drugs to target and break down bacteria. Because if they have something called peptidoglycan, it's a type of carbohydrate, but it has protein in it. The drug can recognize that and inhibit any more like growth or structure, like increase in structure of the peptidoglycan. So the bacteria will die because it can't protect its cell wall or build a cell wall. And then finally, if you guys have probably covered this, hopefully, we have like more modifications. So if it's missing like an oxygen or something, it's something called deoxyribose. And the ribose is just a name for a type of sugar. But does deoxyribose actually sound familiar to you guys? If I say deoxyribose nucleic acid, DNA. So yeah, deoxyribose is a type of sugar that's in your DNA. So like I said, it's drawn linear, but can exist as a ring. We classify it based off its functional group. And I just named these guys. I don't expect you guys to know these. These are just a chemical name, but they have this thing called a carbonyl group, which it's not because everything's linear. This is a carbonyl group. So carbonyl is a C double O. That's for future reference. But It's just classified based off your functional group, and you number your carbons. So that's how you know how many there are. So you can have a pentose, a hexose, you can have more than that, but typically they're these guys. So pent means 5, and hexa means 6. So pentose is 5 carbon, hexose is 6 carbon. And you can even classify bonds. This one's a little bit harder, which I'm not going to explain, but you guys will learn how to do this like in high school. So yeah, don't worry about it. And then carbs are also involved in photosynthesis and cellular respiration. So we do cellular respiration, plants do photosynthesis and cellular respiration. And I'm going to explain like the general process in a future lecture. So don't worry about that. So lipids, they're fats, well, and oils. The delineation between the two is fats are solid at room temperature, oils are liquid at room temperature. So like olive oil versus like butter. So carbohydrates, I told you what the main formula was. We all like now you know it's like it has to be CX in parentheses H2O X. So because 6 like C6H12O6, right? That is a standard formula, and then you can add modifications like adding peptides, adding other functional groups, or you can remove things altogether, and it changes whether or not what the behavior. Well, it changes what the behavior of the sugar is going to be. Lipids are like the weird bunch of macromolecules because they don't have a set structure. The only thing they have in common is that they're hydrophobic for the most part. They do have polar, I'll explain what that means, but they're hydrophobic, which means they don't like water. Lipids are typically made up just like carbohydrates of carbon, hydrogen, oxygen. That's what the CHO mean. So they have carbon, hydrogen, oxygen. Some of them have P, which is like phosphorus. So that's called a phospholipid. But the reason why they're so hydrophobic, meaning they hate water so much, is that they have lots of carbon to carbon, lots of carbon to hydrogen bonds, which the water doesn't like, because likes dissolve likes, and water is not like those type of bonds. So it just doesn't really want to mess with them. And so because of that, it gives like the hydrogen, it gives the lipid some like abilities to form membranes. So what happens is they have a polar bit and a non-polar bit in membranes. So what I mean by polar is a property of a molecule where it likes to interact with water. So it has, like when we're talking about electronegativity, it has like a little bit of electronegativity, like more electronegativity in atom compared to the other. So it's like pulling electrons more than the other. It's not sharing nicely. It's not being like an equal share. Carbon to carbon bonds will share electrons nicely because they're both carbon and they have the same fortitude. And it's kind of the same thing with carbon to hydrogen. It's a weird explanation for that, but it's the same idea. So these guys are, membrane. This is a membrane. This is a polar head, and then these are hydrophobic tails. You guys will definitely cover this if you get into like organelles and stuff when you're in middle school. So this is polar because this is a phospholipid head. So there's a phosphorus group on here. And so this interacts with water, but water doesn't like the tails. So the tails want to interact with each other. And it's something called the hydrophobic effect. So that's what happens. And these tails, they're called fatty acid tails. So fatty acid tails are made out of long chain tails made out of carboxylic acid. The carboxylic acid is not a fun thing to play around with because it's an acid. But what happens is your body and other organisms, what they do is they do this process called a condensation reaction. That's the general name. There's a specific name, but we won't bother with it. But it condensates the tail to a backbone so that it doesn't hurt the rest of the organism. So that's what happened here. It condensated the tails to this backbone that's attached to this phospholipid head. So this is an amphipathic molecule. This means it has a polar head, non-polar tail. So it likes water here, it hates water here. It has both behaviors. But yeah, non-polar, like polar we have in membranes. Non-polar is for insulation and energy. So it's like storage fat. It doesn't want to interact with water. And then the reason I say they don't have the same, like not all lipids look like this, because some lipids can look like this. And this guy's called cholesterol. And cholesterol actually shows up in this membrane. It's like acts like a little spacer. If you guys have gotten like braces or something, it's like those little elastics they put between your teeth to space it a bit. So that's what these guys act. They act as spacers. And so cholesterol is a 4-fused ring, and it is hydrophobic, which means these rings do not like water. They don't want to interact with water at all because the bonds aren't the same. but it does have this little OH, which means this oxygen is pulling on this hydrogen's electrons and it acts polar. So this means it can interact with water just a bit, but not enough to make it fully not hydrophobic. Does that make sense? Thumbs up, anyone? Is it okay? Okay, I'm gonna take it as everything's okay so far. But yeah, and then as far as more lipid stuff, you guys have heard about like saturated fats and unsaturated fats, right? Like when you're eating food and stuff like that, you'll sometimes see, oh, this says unsaturated fats, not like saturated fats, that sort of stuff. Well, basically that has to do with like lipids and their hydrogenation level. So how many hydrogens? It's a fancy word for saying, ooh, this has lots of hydrogens, or this doesn't have a lot of hydrogens. So if it has a lot of hydrogens, like fully, like these tails are covered with hydrogens, that's called saturated. If they're unsaturated, they have kinks because they're missing hydrogen. So that's what this diagram here is showing. There's a kink here because we're missing hydrogen. And so because we're missing hydrogen, this carbon decided to say, well, I'm missing a hydrogen, so I'm going to bond to my neighbor, so that it causes this little kink. And that's really important when we talk about membranes, because it allows for class. Yeah, it is. I think it's an hour and 30. But yeah, if you guys have to go, don't worry about it. I think we are recording this, so but yeah, it's like a kink tail, and that's really important for membrane fluidity, which means, because it's like having a bunch of people in a room, like all packed like sardines. If someone kicks their leg out, right, that one person has more space to spin around. Like if you have a bunch of people all spinning in a room and they're all packed together, like they don't have a lot of space individually, right? But as soon as someone kicks their legs out and starts jumping around and starts spinning like this, that one person has more space, right? So that's basically the point of having a kinked tail. It's like you allow for more mobility. So this is just some trends. We notice longer fatty acids have higher melting points. That just means it takes longer for those things to separate because you have more interactions between each other. So it's like holding hands, kind of. You have more people holding hands and stuff. Then, yeah, sorry. If you had like long tails, you have more abilities for, like, if, I don't know how to explain this in terms of like analogy, but it's just like there's more interactions between the tails than if they were shorter. And if they have more double bonds, it's like more people spinning in a room, kicking their leg out. It's like you have more space to spin, and you're less likely to be like in a can of sardines. And don't worry about the naming system. The naming system itself is like confusing, but that was just a fun fact. We do this thing called delta or omega. It's not too confusing, but it's just too long to explain. And they don't really ask that in middle school. For nucleic acids, it has every element that a lipid would have, except now we have something called, we have nitrogen. So nucleic acids are like your DNA, RNA, ATP. So you guys use ATP for energy, right? So the main gist of nucleic acids in general is when it comes to mind, we always say it stores or transmits genetic information, but it's also used for energy. When we talk about ATP and it's other forms, there's a bunch of different forms. But the main component, like for carbohydrates, we said the component was monosaccharides. For lipids, we said that there's not a set structure. We say that it's made up of fatty acids sometimes. So you can say a component is fatty acids and a backbone. So that's a lipid. In most of the cases, not for cholesterol. But for nucleic acids, we say it's made of nucleotides, which are made of a phosphate group, which is here, and then a ribose sugar, and then a nitrogenous base. And so deoxyribonucleic acid is DNA, and then ribonucleic acid is RNA, and you have different types of RNA. DNA has 4 types of nitrogenous bases. So that's this guy, the thing in purple in this diagram. And that delineates what type of nucleotide it is. So it's kind of like, that's why we say it's ATCG. We're naming those different nucleotides based off its nitrogenous base, if you guys are ever wondering that. So A is adenine, T is thymine, cytosine is C, and guanine is G. RNA is similar, but instead of having thymine, which is a T, it has U, which is uracil. And you have different things called purines, pyrimidines. Basically, purine has 2 rings, pyrimidines have 1 ring, and because of the shape, because of the 2 shapes, they're complementary, which means that they want to be accepted by each other. So it allows for different bonds to form, and that's why you can have like something like a double helix for your DNA, which is kind of cool. And you don't have to worry about the linkage, but that's just what they're bonded by. Like I said, that carbohydrates were formed by glycosidic bonds. Lipids, it depends on what type of lipid we're talking about, but for the most part, we say that they're esterified to the backbone. So that's the fancy word for a condensation reaction for a lipid. You guys don't have to worry about that. But a phosphodiester bond is what bonds each of these nucleotides to each other in a DNA. So it goes from this phosphate to the other sugar, and it just keeps going. Like that's why you get the helix, because it's a shape thing. Proteins. So there's a reason why I did from nucleic acids to proteins, and I'll explain why in a few slides. But proteins in general are made of carbon, hydrogen, oxygen, and nitrogen. When you start getting really specific with proteins, especially in high school and in university, they bring up sulfur and phosphorus. So those two are really important as well. But for general understanding, I would just concern myself with knowing CHON for proteins, because otherwise then you get confused with nucleic acids, and it's not fun. But proteins, so we said nucleic acids are made of nucleotides, and we said carbohydrates are made of monosaccharides. Proteins are made of something called amino acids. And so this right here in the upper right hand group, I know my directions, are it's an amino acid. So we have something called an amino group, an alpha carbon. You don't need to know what the carbon's actual name is, just a carbon, but it's called an alpha carbon for if you wanted to know that. And then this is a carboxyl hydrogen, carboxyl group. This is the hydrogen, and then this is a side chain. And you form a bond between this nitrogen and the carboxyl group of a different amino acid. So right here. And different characteristics come from this R chain. That's all you need to know. And then basically those different properties cause different levels of folding. So it's kind of, I wouldn't say like an origami, but you have different levels of getting it from a flat piece of paper to a folded, like a kind of like a taco folded paper, and then you like shape it every which way until you get this really big 3D structure. That's basically a protein. So you have primary, secondary, tertiary, and quaternary structures. I'm not going to really elaborate those different steps, but you do cover them, I think, more towards 8th grade, 9th grade. But primary structure is just the string of amino acids that are all bound together, and secondary is how those interact with each other to make it like a first type of folding. So it's like taking a flat piece of paper and folding it over once. And then tertiary is getting a third, like a 3D shape. So that's like making like a crumpled paper ball. And then a quaternary structure is taking a bunch of paper plain stuff, and then jamming it all together. And so those different components all added up together make a quaternary structure. Proteins are a really diverse group. So like we see them a lot in enzymes, which we all use, which are really good because they help catalyze reactions, which means they help with like reaction efficiency, and making sure that things need to go where they have to go. They're in your we have channel proteins within your cell membranes, which are really important because that allows for stuff to come into your cells and out of your cells. You use proteins for locomotion, so like your muscle movement, that's all proteins, and because your muscles. And then you have like in your skin, right? You have elastic factors, so you have collagen. Your nails have keratin; those are all proteins. And also like insulin. Insulin's really important, right? Because it's like a sugar. Like it manages your sugar, right? So insulin's also a protein. And then like DNA, I put DNA polymerase. Like DNA polymerase helps with your DNA replication. We'll talk about that later. But yeah, that's an enzyme. You can tell an enzyme is an enzyme by ASE, the ACE part. Just fun fact. But yeah, all those things listed at the bottom, those are all proteins. And there's like so many different types of proteins. They all have really fun names. But yeah, they all do a bunch of different stuff. They're probably the most diverse macromolecule. Yeah. So this is just an idea of different structures. Like I said, amino acid chain, this is primary, it's like a flat piece of paper. Secondary structure, you can have two different ways of folding it, but basically it's like taking a paper, folding it taco versus like not, like I have no idea what they call it anymore, but like the like folding it like the short way or long way. And then tertiary is like the crumpling of the thing, and quaternary is like a bunch of crumpled bits of paper jammed together. So how do they work together? I think you guys know what this is. This is a ribosome. So this has RNA and protein together. So RNA is a nucleic acid, protein is protein. They all interact with each other. This is your membrane, which is actually kind of cool. So your membrane is not just, like I showed you, those little tails with the little red heads. Those are phospholipids. Your membrane's not just that. Your membrane has these blue things, which they're not actually blue in your body, but they look kind of similar. They are channel proteins, and then you have other proteins that do a bunch of different stuff, and then you have these little chain things called glycoproteins, which is a protein with carbohydrate on top of it. So all of these things, they all interact. All these macromolecules, they all interact with each other for like cell recognition, cell signaling, cell transportation, nutrient transportation, like everything is all, they all work with each other for that sort of stuff. So that's why I put down like taste receptors, insulin receptors. The fluid mosaic model is just what we call this model of the proteins and the fats and the fats, lipids and carbohydrates working together. And then this is just a mechanism for epinephrine. So epinephrine is a hormone, and what happens is it binds to this protein here, which sets off this entire reaction. Then you have ATP, which is a nucleic acid, and it forms this other nucleic acid, and it's like everything is like, it all combines together, and it's really, really cool. So it's not just one macromolecule doing one thing, they all work together. So like I said, like macromolecules, they all work together, and like they're all together in food, plants, they're in all biological systems. So that's just the gist of why we bring them up, because they're such an important factor for just not only our cells, but like bacteria, plants, things we get energy from. So the central dogma, this is why I did a certain way of listing the slides in the slide deck, but you get this thing, central dogma of biology just explains how proteins are made. Why does DNA do what it does? What's the intermediate step? What like allows DNA to be, because we said DNA stores and transmits genetic info, but how does it do it? Where does it go? Is it possible for something to go back to DNA, like if it was an RNA? So basically scientists came up with this thing of saying, well, DNA is what's stored in your cells, it's your genetic information. It can get transcribed. So what it means is it's being moved into a different form. It gets moved into a different nucleic acid. So it's kind of like moving it into like shorthand form, because DNA has a lot of stuff going on in it, especially for us. We're called eukaryotes and bacteria is prokaryotes, right? So like for multicellular organisms, you have a lot of stuff going on in your DNA. It's really lengthy. mRNA or RNA in general is like the short form sort of thing. So it's like taking sticky notes. So you take these sticky notes and then you take it and you translate it. So it's like changing the language. You change it into proteins. You change it into amino acids. So that's the central dogma. It's DNA transcribed to RNA, translated to protein. So then that begs the question of, is it possible, is there steps that can go back and forth? Can we take RNA and go back to DNA? And the answer is yes, we can. It's called reverse transcription. So in that case, it was good that scientists didn't give a crazy name, because if it was transcription to get to DNA to RNA, it was good that it's reverse transcription to get RNA to DNA. And reverse transcription is really important for like a bunch of different like research methods, especially for when we're talking about like cloning and things like that. Because, for example, in human DNA, I'll explain this like I think next week. But in human DNA, you have all these excess nucleotides that don't really do anything, because they're acting as spacers. They're trying to make sure the DNA is protected. But when you get to RNA, those extra bits are cut out in the RNA to get something that's more condensed, short form, like I said, like a sticky note. So if you're taking a condensed RNA and changing it back to DNA, you basically get a short form DNA without the extra bits, and it's much easier to analyze in terms of research. But that's just a side note. So does that make sense to everybody? Because now we're getting into the application activities. I just want to make sure I have enough time. Yeah, I do. We good with that? Any questions? No? I'll ask, let's see, I can ask a couple questions. So based off what you just learned, what's the most diverse macromolecule group out of the four? Is it carbs, lipids, nucleic acids, proteins? What do you think it is? Okay. It's proteins, right? Because we said that proteins can do a bunch of different things because they're from nucleic acids, and you can do a bunch of different, like when nucleic acids get transcribed and translated, there's different ways that the protein can get folded. There's different functions it can do, right? It can do, it can be an enzyme, it can be a channel protein, it can be like a cell transporter, it can do all these different things, right? It could be a peptide hormone like insulin. Peptide hormone is just a fancy name. So that's that. Let's see. And then, yeah, no, I'm not going to ask that question, but I'm just going to get straight to the application activities. Did the past few slides make sense to everybody? You can just put in the chat yes or no. Okay, I'm just going to take that as a yes. If you guys have questions, just let me know. So at the end of these things, I do a little bit of application activities just to make sure you guys understand. Okay, you're a little confused. What were you confused? If you have like specific questions, so just to recap, you have 4 types of macromolecules, because these questions fall into it. You have 4 types of macromolecules. Macromolecules are in you, me, and a bunch of biological systems. So plants, food, all that good stuff, right? So like we use macromolecules. So your 4 types are carbohydrates, lipids, nucleic acids, proteins. Yeah, it's okay, no worries. I'm just recapping really quick. So you have carbohydrates, which are made out of monosaccharides. Those are sugars. So sugars are used for energy, energy, structure, storage. That's typically what they are. Sometimes signaling, but I'm just going to tell you it's energy and storage. Lipids are like fats and oils, and those are used for energy and like storage of energy. And they're also used in your membranes. So your cells have something called membranes, which hold the cell together on the outside. And then you have nucleic acids, which are bits, like in general, bits of, like they basically hold your DNA, like they hold your genetic information and store it or allow it for it to be made into protein. And protein was the last one. Protein is the most diverse macromolecule. Protein is basically the end product of your DNA. So your protein does a bunch of cellular functions, and it's really important for you to have because you need it for different reactions and you need it for different building blocks for your body and stuff like that. So that's the 4. And basically when I went through those past few slides, I just kind of said what those individual building blocks were for those different macromolecules. And then I gave like a bit of examples. So when this gets released, when the slides get released, I think it'll make a little more sense. But I'll just do these application activities and really quickly and just say how I would solve it. So for this one, you were given a bottle of triacylglycerols, which have fatty acids. So I was just kind of telling you it's a lipid. And then you were given a bag of sucrose. So I said it was table sugar. And so the main point you should get from carbohydrates is that sugars are carbs. So carbohydrates. Then you pour each of these substances into a bowl of water, and then you see that one dissolves and the other doesn't. So why do you think one would dissolve and why wouldn't it? Based off the idea, if I told you that likes dissolve likes, and what was the most, so let me ask you a question. What was the most common property of lipids? Did I say they liked water? Did I say they hated water?
I think it's both.
So yeah, you're right. There are some types of lipids that like water and don't like water. Those are phospholipids. But in general, if I was just saying, because lipids don't have the same structure, right? Do lipids like water in general, or do they not like water? Like I know what you're talking about, but like, do I say all lipids like water? Is that a common thing? Because what about like cholesterol? What about olive oil? If you pour olive oil, if you pour olive oil into water, does it like it?
No.
Yeah, no, right? So in general, lipids don't like water, but you're right. You do have exceptions that are like they like water and don't like water. So those are in your membranes. And the bit that likes water isn't actually a lipid. It's a phosphate group. So the actual lipid, the fatty acid, which is the carbon backbone, the hydrocarbon backbone, the one that has the hydrogens bound to the carbons, like the long squiggly tails that I showed you, those are called fatty acids. And those don't like water because the likes don't dissolve likes dissolve likes don't dissolve dislikes. That's what I'm trying to say. So if it's the same sort of substance, they want to dissolve each other. If it's not the same, they don't want to do it. And it's based off of interactions. And I'll explain why it does that in the chemistry bit. So sugar has similar interactions, the bond interactions, and so that's why it dissolves. But the fats can't do the same thing. That's the difference. And that's what I just put here. And then this is a question about when I was telling you guys about, so like this is whoever was saying that they both liked water and disliked water. Yeah, these guys do. So this little blobby circle oval thing, this is a phosphate group. It likes water, but it's not, it's part of the lipid, but it isn't the fat part. So the lipid part is the tails, these little squiggly leggy things. So this question is asking if they're amphipathic, which means both water loving and hating, if they had more double bonds. So when I said picture of people packed in a room all together, but then someone sticks their leg out and starts spinning, that person makes more space for themselves, right? So if I have more people jammed in a room together, right? And I increase the temperature, and let's say increasing the temperature makes them space out more. If I have people in a room who are packed like sardines, right? There's a lot of people all close together, no one's sticking their leg out, and I increase the temperature, but I have a separate room and I have a bunch of people with their legs sticking out and spinning around, and I increase the temperature there, who will spread out more faster? Or who spread out first? The people who are really close together or the people who are already spinning around, kicking their leg out, making more space? Anyone? That's kind of what this question is asking. Unsaturation versus saturation. Unsaturated is the people with their legs kind of had a kink, like they're jumping around on one foot, spinning, and they have more space. And then people who are like packed like sardines are the ones that are saturated. So they have no space to move. Which one do you think it is? That question kind of makes sense. If it doesn't, I can explain the answer. But basically, so this is unsaturated. See, there's nothing that has kinks. No one's sticking their leg out, no one's spinning around. They are spinning, but they don't have a lot of space for each other. Whereas here in this one, these guys are unsaturated. So they have way more space to spin around, stuff like that. So in general, when you have higher temperatures, you're putting in more energy to a system, and that makes it really, like, really agitated. So it's like, let's see, trying to make a good example. I can't think of a good example right now. But basically what happens is when you increase the temperature, you give more energy to the system. And so it's like having a box on the table and you have coins in it and it's just sitting there. That's like if it was cold. And then you start shaking it really hard and the coins start moving every which way. That's like increasing temperature a lot. So that's basically what you're doing. You're shaking the membrane so much with the temperature increase because you're putting so much energy. And if they're all close together, they will break apart, but they won't break apart as fast as these guys will if they have kinks, because the kinks make them have more space and they're already moving more than they would have if they were in this scenario, if they're all jam-packed together. Does that make sense? Sort of. If you guys are confused, just please let me know, because yeah, I think I put it here. So I said more unsaturation means you have more double bonds, which means you have more kinks. So more people spinning with their leg out, just rather than being packed together. So you're not packed together like these guys would be. And at higher temperatures, you would have to move, molecules move more, right? So that if you're moving more and you have less packing, your membrane would have to break faster. Okay, cool. So yeah, that is all I have. These are some helpful links if you guys want to read up a bit more. Thank you for coming for the first session. If you guys have questions, I think I can stick around for a couple more minutes. And then I think we're releasing that optional assignment. And then next week we'll have like a mini Kahoot before our next thing, so that you guys get a little bit of a refresher of the material. But yeah, if you guys have any questions, please let me know. Feel free to ask Sherry. Sherry's helpful. She's there for you guys. Yeah, thanks for coming. Hope you guys enjoyed that. Stop share. So, are there any questions for you guys now, or yeah, no worries.
No.
Alrighty.
Good. Hi.
Hi. Shared. So, do you want to stay a couple more minutes, or do you want to just end the thing? Or there's someone.
Julia just came in. I'm not sure.
Oh, hey Julia, do you have any questions? I can stay like a couple more minutes probably, but yeah, I think that was. Oh, yeah, no, she just.
Oh,
Popped out.
okay.
Yeah.
I was
So yeah.
wondering, could you send the PowerPoint to me as well? I could include that in the email. And then
Yeah, I will do that just now. I just had to, sorry, I had to double check to make sure I put everything in because also I didn't want to send the wrong slides. I was looking, I'm like, I think I have the right slides, but yeah. And so I will send you the Kahoot stuff. It's up to you, like when you want to make it. We're just probably gonna do it the first 10 minutes of class. Yeah.
yeah.
I think next week's chemistry, so hopefully should be a bit more straightforward. The problem with like bio is like there's so many terms, but chemistry is way more easier to explain, I think. But yeah. So yeah, I will send that to you.
Is this the only biology class?
Let me double check. I don't think so. Well, that because I'm doing things a bit differently than I did the last term, or not term, sorry, last summer. Because last summer I did 2 classes per week, which I think were 45 minutes each. So this one we just kind of condensed everything. 6 weeks, let me see. I'm getting there. Oh no, sorry, week 2 there's more bio. I'm like jumping ahead to week 3. Yeah, no, week 2 we're doing organelles and cell stuff. So this is where like the macromolecule stuff kind of comes into play, and then a bit of energy enzyme stuff, but mostly I'm not gonna really go too crazy on that one because I think sometimes that's really hard to understand if you haven't really seen it before. And then there's cellular respiration and photosynthesis, which I know you guys in middle school and high school have to do a lot of.
Yeah,
And then
what
I'm just
about
doing,
like, I was wondering, what about like processes like mitosis and meiosis and that kind of thing?
oh yeah, that is where did I put that? I did that more for the genetics part of stuff. So there's I'm doing like Mendelian genetics, and then there's some bit of population, but on it, I think the last time I just combined all of the geez, I'm like blanking, the DNA replication stuff underneath Mendelian genetics, just because sometimes like these things get so confusing. You know, it's just I don't want to throw in like it's everything with bio, like I said, it's like you have DNA polymerase, and then you have ligase, and then you have all these other like different things, and if you hear it all for the first time, you get like kind of confused. So I just don't want, I mean, even this was probably overwhelming, so I'm just like I don't want to overwhelm you know. But yeah, no, it's coming, it's there, it's just I didn't really specifically label it, but yeah, no, it's in my lecture notes. So yeah, just to think about that. So yeah, we good. We good on the, I'll get you the slides and then I'll send you the Kahoot. And if the Kahoot is too long, I think this was an issue last time. If the Kahoot is too long to fit inside the prompt box thing, feel free to like cut stuff in a word, like you can change the question, like the same question, but like change the sentence forming stuff. Because sometimes when I write it like really long and I don't mean to write it that long. That works?
I'll take care of the wording and stuff.
Okay, sounds good. But thanks, Sherry. I don't think anyone else is coming, but yeah. Anyways, yeah, see you next week.
See you next week. Bye.